Malacology ExLENT Workshop. Welcome. It's good to have you.
Hi.
We've got a great presentation lined up for today. So if you have not signed in at the front desk, please do that. You can also use the QR code if it's the same sign-in sheet. And then we have lots of food and drinks over here. Anybody who's in the room, regardless of where you're here, please come help yourselves. There it is. Just sign that and help yourself. Make a plate, get seconds, get drinks. Please eat up. And for anybody who's new to the STEM Center, we have a water fountain over here. You can drink the bottles. And if you need the restroom, it's out this door, down the hall, past the elevator. So, I'm going to tell you just a little bit about our grant really quick. This is our second year of ExLENT, and basically we are working in kind of three parts. So right now we are in the Inspire. So today you're going to be hearing a tech talk from Ken and Nori, who are here from the Malacology department at Bishop Museum. And next spring, we will be taking a group of students out to see their lab and the whole department. So those are our site visits. We call that Inform. And all of this leads up to Immerse, which is an 8-week paid internship. So the internship pays $5,000. Anybody who would like, as long as you're a Kapiolani student, you can apply to do this internship. But the more of these events that you participate in, the better your chances of being well matched with one of our partners. That it's best if you participate. So hopefully everybody has got one of these packets. It's got an IRB, and then it's got a survey. It's got a green survey with some snails on it. So really quick, this is a grant, and we do collect some survey data. And so the IRB form, if you consent to being part of this, there are a few things that I would appreciate you filling out. So we are taking a video of this workshop, and we take photos throughout the thing. If you don't want to be in any photos or you don't want to be in the video, you can opt out of that, and we'll make sure that you don't end up in any of those things. And then please print your name, sign your name, and at the date. And the most important thing to know when we do these surveys and we collect data is basically just that this is voluntary. If at any point you don't want to continue to let us collect data, you can rescind your consent and we will not continue to collect data. So everything is optional. You just have to let us know. And so, that being said, now this is the first page of that survey. So there are a handful of questions, and basically what we're trying to do is ascertain at this point before the workshop where you feel like your level of comfort with some of the knowledge is. So the first question is, I understand what emerging technology fields are. And then you just circle which whenever aligns most appropriately. Second is, I am likely to pursue an emerging technology pathway. The third one is, currently I see how technology fields are applicable in Hawaii. Fourth, I understand what emerging technology jobs, what kind of jobs are available. And I see how indigenous knowledge can be applied in an emerging technology field. So that's the whole thing. I'm going to give everybody a moment to, there we go. Since we've got some folks who are coming in, I'll give everybody a chance to mark their surveys. Make sure that you put your name clearly on the IRB. And on the survey, if you could just tell us, you don't have to put your name on the survey. We don't collect the data in the same way, but if you want to tell us what your degree pathway is, that would be fantastic. And so while everybody kind of catches up on that, I'm going to tell you guys a little bit about Nori Leung and Ken. So I had the privilege of going to one of their workshops at the Ship Museum, and it's just such an interesting field, malacology. I had no idea how many tree snails there are and to what degree they contribute to the health of our entire island ecosystem. And so they're going to be talking a lot about that today. So Nori is doing, I feel like I'm gonna mess up who does what. So I know Ken is working in the Pacific Center for Molecular Biodiversity, and they're both at the Bishop Museum. But basically, if you would like to intern and do DNA and sequencing and microbiology stuff, Ken is gonna be who you wanna be with. Nori is doing really cool stuff with 3D printing shells from extinct species of snails and from ancient species of snails, and they're doing that so that people can take these 3D printed objects and interact with them in a way that won't damage the actual shells themselves. And I think at this point, I'm going to turn it over. Who would like to speak first? Ken? All right. So this is Ken, and I am going to switch to your slides. So give me 1 second. And
all right. Well, good afternoon, everyone. Thank you for the invitation. The other person we didn't introduce, we just make her come along and do work, is Jan Kapiolani. She is our captive rearing coordinator, so she oversees the land snail rearing. Our first slide, I can't remember. Yep, that's the first one.
I believe so, yeah.
Oh, is that your slide? Title slide. Anyway, okay, so as Rita mentioned, we work on land snails a lot, but it's only it's a large proportion of what we do, but we do quite a bit of work. It's basically surrounding the theme of biodiversity, right? As you know, Hawaii is a biodiversity hotspot. We sit right out here in the middle of the Polynesian, Micronesian hotspot. And then there's also the, which extends all the way over, but we also have the East Melanesian island hotspot, right? So those three hotspots, the Micronesian, Melanesian, and Polynesian hotspots make up the Pacific hotspots and encompass some 25,000 islands. And we study land snails simply because that's kind of what we developed over years of working. There wasn't anybody else to work on them, but we also work on a whole variety of things. As a matter of fact, I just isolated some tardigrades from the Northwest Hawaiian Islands. We're getting ready to describe those. Does anyone know what a tardigrade is? Well,
Water bear?
a water bear, sometimes called water bears, right? Exactly. So these are small microscopic animals. They're the only animal known to survive the vacuum of outer space and the UV radiation of outer space. So some people ask, so what, right? Well, the interesting thing about that is as climate continues to change, we're going to need to develop technologies to deal with that so that we can live on this planet still. So for example, some 30% of the world is fed with rice. Those rice areas, right, are going to be inundated with salt water soon, many of those rice growing areas. So what do we need? We can either move rice growing areas, which is unlikely, or we're going to have to develop salt tolerant rice plants, right? Guess what? Tardigrades are also highly salinity tolerant. They have genes that allow them to deal with salt, so we can transfer those to plants or develop plants that are that way. So there's lots of technological applications. And while we primarily work on biodiversity, we use a wide range of technologies ranging from electron microscopy to 3D printing and a lot of genomics technologies. And I brought some of the equipment to show you that. But in general, we always like to start out with the why, right? Doesn't matter what you're doing if the why doesn't really make any sense. And the reason we're doing this, biodiversity hotspots, who knows how you define a biodiversity hotspot? Anyone? What do you think that means? Let's break the word down. Hotspot, right? That's usually where something's the hot spot, that's where it's happening, right? So if we just take this word at its roots, bio, right? Life, diversity, the diversity of life hotspot. So these are places that have incredibly rich biodiversity, but there's another component that makes them qualify for one of the 36 hotspots. And that is that biodiversity is heavily threatened with extinction. right? In other words, there's a high level of endemicity. So for example, our Hawaiian land snails, they're 99.9% endemic, meaning they're found nowhere else in the world except in Hawaii. Yet they're heavily threatened with extinction. So that's what makes them a biodiversity hotspot. When you start looking at the plants and animals, their diversity, endemicity, and then whether or not they're threatened with extinction. Okay? So again, here we are, right out in the middle of the Pacific, right? And we're often talked about as if we're the extinction capital of the world. People use that. So we're one of the biodiversity capitals of the world, but we're an extinction capital as well. And that's because, for example, half of all the endangered species on the endangered species list are found here in Hawaii. More than 500 species. Right? All together, if you take these spots across those 3 hot spots, you'll see about 600 total species. The U.S. mainland only has maybe 1,000 at most. So what do you think the threats are to biodiversity?
Climate change.
Climate change is a big one.
Introduced species.
Introduced species is probably one of the biggest ones here. What else? What does Waikiki look like today versus what it looked like, say, 300 years ago? Okay, what was Waikiki? Wetlands. What's that?
Marshlands.
Yeah, it was marshlands. It was wetlands, right? And then they dredged the Alawai Canal and filled in that and built a bunch of hotels and beaches, right? Completely different. So that's habitat destruction or habitat modification. Obviously, not very many native species that lived there then are there now, right? So those are the big ones, right? We often think of those as the 4 horsemen of the biodiversity apocalypse, right? So overexploitation, overfishing, overharvesting, right? Invasive species, big one here. Climate change, and I'll argue that climate change, while it is big and it's in our face every day, it's a technological issue, right? It's not that difficult to solve. We know what causes it, and we actually have a lot of solutions that we could probably fix it. What we lack are the will to do it and the funding, right? Not the money, right? We have the money, we just don't use it to fund climate change work. We can fix that. So I always like to tell everyone, if I drove up to the parking lot out here with a truckload of money and said that I'll continue to bring a truckload every day until you fix climate change, do you think we could fix it? Oh yeah, no doubt in my mind, we could fix it. Within a few decades, we could reverse most of it, we could make the place better. Right? Same thing with habitat destruction. We can restore habitats, we can do a lot of these. But here's the problem with biodiversity loss. If I gave you that same offer, and I drove up a truckload of money and said, okay, fix biodiversity loss, what's the likelihood that we could fix any of it? Impossible, right? We can't. And the biggest thing that's preventing that is this right here. Like I said, we know what causes climate change. We know how to fix it. But knowledge shortfalls, and that's the really big, the sort of 5th horseman in the back rushing up on us that we often ignore. We focus heavily on these, but we're not focusing on this as much as we should. We have these large knowledge gaps, particularly when it comes to biodiversity conservation, that we're unable to fill, right? And we can't address it. It's like if you said, okay, go out and save X number of species. I couldn't even begin to tell you how we do that. We only know a tiny fraction, right? So these are these knowledge gaps, right? They've been quantified or at least qualified and discussed extensively in the literature for the last decade or so. The big one, we call it the Linnaean shortfall. You know, Linnaeus was the father of modern Western taxonomy. He's the one that decided, like, oh, we should have a genus and species for every name, the binomial, right? We still use it today. So this is essentially this shortfall in we haven't discovered all the species. We don't have names for everything, right? And then distribution, so this is the biogeographic shortfall. So if we don't know what's out there, we can't possibly tell you where they are, right? So Wallacean, that's Alfred Russel Wallace, named after him, the father of modern biogeography, right? So those two together, don't know what's out there, don't know where it's at. How do you even begin to conserve what you can't find or identify? And then all of these follow from that, right? So we can't tell you how many they are. We can't tell you how they evolved and who they're related to. We can't tell you about what their tolerances are. So, okay, great, we know climate change is going to be a problem, but how do we know what's going to survive higher temperatures, right? Drier conditions, those kinds of things. The traits of them, how they interact with one another. This is a big ecological one, and the father of invasion biology, Elton, kind of came up with this idea. And then the biggest one now, right, where filling in those knowledge shortfalls and things, are in this one called the Ostromian shortfall. And this is actually named after a former Nobel Prize winning economist, right? So it was an economic theory, but applied to why do we still have these problems in biology and the conservation? It's because we lack evidence, we lack the information to make decisions and develop policy, which affects our ability to get funding and all of these other things, right? So I can guarantee you I can go to most audience like this, and this is probably a fairly well-educated audience, right? We're speaking to sort of our peers here and that you guys attend classes or educate, but if I go almost anywhere in the world and start talking about these things, most people have no idea, right? Rita mentioned that she didn't realize how much diversity went on in the Fish Museum, but it extends way further than that, right? People don't realize how many land snails are here. Most people have never seen a tardigrade. And again, most of your daily life isn't affected by that, but our ability to conserve and manage all of our resources is highly dependent upon the broader community having an understanding of these things. Even if they're not experts, we don't need everybody to be experts, but they have an understanding. Why? Because when you vote, who do you want to vote for? Do you want to vote for the guy that's going to allow another hotel to be built and drain the water from the mountains? Or do you want to vote for the person that's going to try to take an understanding of this and figure out how we more sustainably live on this tiny island, right, without depleting its resources? These are really important issues, right? So, there's roughly 10 million species on the planet, we think, right? It's sort of this best guess, right? How do you know what you don't know, right? So, we do know that we know of about 2.5 million species right now. We've got names for 2.5 million. A lot of fudging in there because some of them may be synonyms. We may call, one thing two different names. And in some cases, we may be calling 10 things one name. So there's a lot in there, but there's roughly 2.5 million, right? And we like to say at the museum, right? Just see on the edge of Jan's shirt, it says 25 million stories and counting. So species on this planet, including us, are like libraries in the library, our books in the library of life. Each species tells a story about life on this planet after millions of years of evolution, right? And so that's what we have here, right? So, but imagine a library, right, that is got 10 million volumes, but you can only find 2.5 million of them. And you don't know how to read most of it. It's all in different languages. And some of them have the pages ripped out that are missing, right? It'd be a really difficult library to use. And that's kind of what we're dealing with. The biggest problem is we have 7.5 million untold stories. So we can't possibly understand our place or how to manage the resources when fully 75% of biodiversity is completely unknown to us. Okay, so this is a big problem. So each one of these blocks, right, represents about 33,000 species. And here's the thing, where do we think most of those are going to come from? Do we think it's going to be birds and mammals? No. Most birds and mammals we've discovered. There's a handful of new ones every few years, but most of those are discovered. And you can tell from these colored blocks. So this blue, that's arthropods, primarily insects, right? Makes up 51% of all the animals on the planet, and that trend will probably continue. This orange color, that makes up the mollusks. That's the group that we work on a lot. That's snails, right? Octopus, squid, clams, scallops, those kinds of things we like to eat, right? We've got mushrooms here, the fungi, a bunch of other invertebrates, and then plants down here in green. Humans, birds, right? So mammals, birds, fishes, reptiles, amphibians. The things we think of when we think of animals, right? When the first thing that probably pops in your head and pops in most of your head when you say animal, you think of some sort of mammal or bird. Maybe a fish, if you know what your background is. But it's two little tiny blocks compared to everything else we know. But here's the problem. We spend some 80 to 90% of our research and conservation funding on these two blocks. Do we think that's probably a good strategy for saving biodiversity, or more importantly, saving the place we live? Probably not a good idea. Right? And just to give you an example of how this manifests itself. So Linnaean shortfall, right? Sometimes we think of Linnaean taxonomy as this sort of esoteric thing that only scientists use, right? But it's really important, because we have to be able to communicate what's going on. And here's where the problem is, right? So this is a graph showing numbers of millions of species on Earth, all the way up here at 10 million. We're down here at about 2.5 million not too long ago. And then the years on the bottom. So that black line is what's called the species accumulation curve. That's the rate at which new species are described each year, right? So there's somewhere between 16 and 20,000 species described each year by science. And at that rate, If we keep that same rate, we're looking at 2,500, right? The year 2,500 before we reach 10 million. And that's not really a big problem if we had this 0.1% per decade extinction rate. That's the background extinction rate, normal, right? And even it wouldn't be too bad. I mean, we're still going to lose 20 some odd percent by about 2350 before we discover them if the extinction rate's 1%. But here's the reality. We live in a time when the extinction rate varies somewhere between 5 and 10% and as high as 14% per species in Hawaii. And at that rate, you can see where these curves intersect, right? We're looking at about 2,100, somewhere between 2,100 and 2,200 before we lose somewhere between 65 and 70% of all species on the planet before they're ever discovered. This is a huge problem. And this is where technology comes in, right? Because we use traditional taxonomy, which sometimes can take decades. And the reason this is, look at this. This is the 21-year shelf life. So this is a study from 2012. They looked at plants, fish, tetrapods, insects, algae, bacteria, fungi, other invertebrates. These are the average rates of shelf life, meaning that from the time it's discovered until it's given a name. It's 21 years across all taxa, 74 in plants, 9 in fish, 11 in tetrapods, 316, right? I mean, it's just simply because some of them are hundreds of years, right? And that's because they sit in a museum somewhere and there are no experts. I mean, if you make up 51% of all the organisms on the planet, how likely is it that there's someone to be an expert on all of those? No one, right? And then we lack those expertise. But we can't train that expertise fast enough, right? We need to do that. But we also need to bring technology to bear on that, right? We've got to basically steepen this curve. The rate at which we discover new species has to go way up, right? And then we've also got to use technology and our understanding to flatten those curves out, right? To slow extinction and increase the rate at which we discover them. And those two go hand in hand. So one of the technologies that we're heavily involved in, like Rita mentioned, I run a biodiversity lab. We use genomics tools. Also work in malacology and then a bunch of other invertebrates. But one of our big projects that you could potentially be involved in if you're interested is called the Hawaii Barcode of Life. And our objective is essentially to create a short DNA barcode, looks sort of like this, for every species in Hawaii. And that's part of a larger initiative called the International Barcode of Life, which is doing the same thing around the globe. And there's several of these initiatives. So we are on our way, but we got a long way to go. How many species do you think there are in Hawaii that we know of? Wild guesses. There's no wrong answers because we don't really know. But we have a good guess. Anyone know? 5,000? 7,000? 10,000? 15? 20? 25? 30? No? No guesses? So there's roughly 26,000 species described from Hawaii right now. We estimate there's another 10,000 yet to be discovered and described. So somewhere around 35,000 species probably. To put that into perspective, Hawaii is 0.2% the land mass of North America. 0.2%. And we have more species, plants, animals, right, fungi, than all of North America. Yeah. It's enormous, right? Put it in that perspective, it's crazy. So give you sort of more take-home snails that we work on. There are 759, 60, 61, 62, somewhere. They keep going up. Some 760 species of Hawaiian land snails. North America has about 1,000, right? So we have an incredibly rich biodiversity of land snails here. We also have some incredible birds, right? And again, other projects that we work on, we call this wildlife forensics, using technology to better understand and manage wildlife. So we've got a lot of endangered birds, not only forest birds, you've got our pueo, as well as some of our storm petrels and our albatross, right? And we use these technologies, right? So when you look at, say, an albatross, for example, a Laysan albatross, it's not always easy to tell which is the male and the female. Why? Because they look similar. You can sit out there all day and watch them dance, right? And the males dance to attract the female. And so then you say, well, I think that's a male and that's a female. But guess what? You'd be wrong most of the time, at least 30% of the time on Kana Point. Why? Because 30% of all pairs at Kana Point are female-female pairs. And we wouldn't know that without genetics, right? And that's because there's a shortage of males, right? There's more females than males, but they still need to mate. So they do this really cool thing called sexual reciprocity. So 1 year, the females get there, and a male, the early coming back males that already mated, they're paired to someone else, they haven't mated with their female yet. They show up early, they mate with one of the female pairs, and then they mate with their other pair. Then the female that's mated with lays her egg, and they take care of it together because lays and albatross require two parents. One has to fly out to sea for weeks at a time to find food and bring it back. So someone has to stay with the egg. So one of the females stays there with the egg, the other one flies away. The next year they swap. It's crazy, right? Next year the other female that didn't get to lay the egg last year, that's the only way evolutionarily it makes sense. Because otherwise the other one would just be sacrificing. It would be absolute true altruism. But what they're doing is they're essentially hedging their bets, that they each get to even out and mate each year. Rather than if they just competed for males, a lot of females would get no mates, right? So it's really interesting, but you can only get at those kinds of things. You almost can never get at that by just watching them, by traditional wildlife study methods, right? This requires genetics, and so we use genetics to sex birds, we use it to sex bats, we can sex humans too, if need be, but it's really nice because not only that, it allows us to do it non-invasively, right? So these are FTA cards, a technology where you embed a chemical buffer on there that preserves DNA. So you put a little bit of blood on it, or we can do it directly from their feathers or from their eggshell. So you get at that information non-invasively, not without harming the animals. Some of the technology I brought to show you, we won't set it up, but if you're really interested in next generation sequencing technologies, this is a little video going here of this machine right here running and sequencing. So I always like to remind everyone, especially younger generations, the Human Genome Project started in 1985. It took 15 years and cost $3.5 billion. I can do that overnight with this for $1,000. Right? Technology is incredible, right? So this machine you see up there, it has a little flow cell that has membranes on it with pores in it. And as a molecule of DNA passes through that, it changes the voltage. It's measuring that voltage and telling you what the DNA sequence is as it's passing through it. And it can do thousands of base pairs per nanosecond. So what you're seeing here is 30 million reads, right? Right now it's telling you that there's roughly about 2,000 pores on this. As it gets used up, each one of these represents a pore. It's telling you how many pores are available, how many are being sequenced, how many are recovering, how many are inactive. It's real-time sequencing. So I can look at this as it walks through that graph over there and say, okay, well, I know I need about 1.2 gigabases of data. Once it gets to that, I can just stop it. I don't have to run forever. And this is real powerful because it allows me to customize and really tailor the sequencing methods I use, right? These are the same machines that were used heavily during the COVID outbreak, and they still are. So wastewater is screened with these machines because they're economical, they're portable, and they're really amenable to a lot of flexible different technologies. So this is the first generation. This is the next one, right? So this is the same thing, just a little bit bigger. And then we go up to the big bear of them all. So these have a couple thousand pores in them to sequence. Each one of these has 5,000. So you get 10,000 total in this. So you can do multiple genomes. And we're using this to do everything from looking at gut microbes in snails and birds and other animals, as well as environmental DNA in our forest, soil microbes, but also, to sort of investigate and study land snails broadly, right? And that's what Nori primarily leads. She's the senior malacologist, so it's the person that studies mollusks, right? And she's been leading this effort for more than 20 years to study land snails. So she's going to tell you a little bit more about that, and then I'll swap off and tell you a little bit about some of the other technologies we use to do it.
Thank you.
Yep.
All right, so this will be very quickly because we want you guys to do some activities instead of just listening to us talk the entire time. So yeah, like Ken was saying that we have about 759 species, 99.9% endemicity, which means 756 of these species are only found in Hawaii. So once they go extinct, there's no place on Earth that can receive these islands with these snails. So when we call them Hawaiian land snails, they really are our land snails. And here's a misconception that all of our land snails are in trees, but actually it's not. We have snails from the rocky rubbles of the forest floor and leaf litter all the way up to the highest trees and everything in between. And so we don't have any earthworms in Hawaii that are native. So our Hawaiian land snails were pretty much our detritivores, and our conservation managers are recyclers. So they're the ones who are breaking up all of the animal waste, the plant waste, pooping out fertilizer packets, basically rich fertilized soil, so that plants can reuse it again. And none of our land snails eat live plants or live seedlings. So they're not like any of the invasive snails that come from the mainland that eats pretty much everything in our garden, all of our native plants and such. So our native snails actually really keep our forests, our streams, all intact by what they do as ecosystem services. They also have, in an ecological and evolutionary term, they have really cool different reproductive strategies. Some of them have male and female counterparts in the species, but the majority of them are hermaphroditic. So they both have both the male and female reproductive organs. And that also aids on how they're able to colonize a lot of different isolated areas. You don't have to find an opposite sex mate. You can just have to find a like species to get babies. So that is a very helpful reproductive strategy if you're trying to colonize very isolated areas. They have also different ways of laying eggs. So some of them lay eggs outside the body, right? And then they usually lay eggs in areas where it can be protected in like shrubs, leaf litter, logs. But many of them are actually what we call ovoviviparous. So it looks like they're having live birth, but technically the baby hatches in the mama shell and then comes out live. And that's also very important because it protects the keiki in the mama shell, so that if you are trying to be colonizing a lot of areas, this is a very good strategy to have. One thing cool about our snails is that there are different shell shapes, so it's just not one single type of snail that came to colonize these islands. If we have the whole entire land snail tree of life, they come from all parts of this tree of life of land snails throughout the entire world. And later on, we can show you some of these snail shapes. And like Ken said, we did 3D prints. So the majority of our snails are really super tiny, right? So like this one is the size of basically like a rice grain. And if you guys can see that dot is right there. So most people are like, oh, it's brown. It's like not that great. But when you do a 3D print, this is actually what they look like. All the really cool ridges and things. So I can pass this around so that you guys can take a look at it. So these are some of the uses of technology that we use, not only for educational outreach, but what we're trying to be doing to show people how to be identifying different shell shapes and taxonomy and trying to figure out what species there are. So after we do this lecture, we'll have 3 primary sections that you guys can take a look at. I can talk to you more about some of our snails as well as the cultural importance. Ken over here can be showing you on how to be identifying some snails, so we can show you the field guides that we have. And Jan also brought some of the live snails for you guys to be looking at, and to be telling you some of the conservation status and threats that we have for our land snails. So in Hawaii, and actually a lot of places in the world, some of the ecosystem services include food, right? So this is a native forest bird that unfortunately last year it was now dubbed as extinct, and it's a very sad case. It was also known to eat snails. So there's also like possible correlation that yes, habitat destruction, mosquitoes, but what if their food source was dying, snails? Because people don't really care about snails, so they never really understood or studied or how it may have impact our ecosystem. We also have a very cool endemic caterpillar. It's carnivorous. Most caterpillars are non-carnivorous, but this one is. And the species name is called Molluscivora, so it's an eater of mollusks. And there are also a whole bunch of different publications out there that when you're removing invertebrates, particularly like snails like in Hawaii, it changes the nutrient dynamics of the forest. So we're basically taking out all of the healthy things that are keeping our trees, our plants, our shrubs, our ferns healthy, because we're removing these snails because they're basically going extinct. Our snails also have a huge cultural component. So while a lot of places on the mainland are saying, ew, yeah, gross, snails are gross. In the Hawaiian culture and a lot of parts of the Pacific, snails and mollusks were important for the society. So a lot of the marine shells, they were used for food, but they were also used as tools. So this is a he'e lure. So the Hawaiians use a kauri, a snail, to capture an octopus, a he'e. So you use a mollusk to capture capture a mollusk. This is Queen Okalani's Agatanella shell-lay. We could not bring her lay because it's very delicate and such, but we did reprint a lay at first. So you guys, so this one, even 2-year-olds can play with it. Versus that one, you can't even touch it. So these are some of the things that that we have used and created for education outreach. But if you have any ideas as a graphic designer, graphic artist, we would love to work with you guys because there's a lot of content that we have that we haven't developed for the public yet, and we really need the talent and the artistic talents that you guys have. And then here are the many different land snail names in the Hawaiian dictionary. So a lot of the snails were found in the realm of the gods, up in the Mauna, right? So these were special snails, snails that sung, that were told in love stories and were talked about as good on men. So like after the rain passes, listen to the land snail sing. So in the Hawaiian culture, it's also incredibly important. And also because in Hawaii, most of the knowledge is oral. There's no pictures, there's no photographs, there's no tapestry. So once a species in Hawaii goes extinct, whether it's a plant or an animal, the Hawaiians no longer have anything physical that they can refer to. And that's very hard because we have such incredible songs and stories, but if we don't have anything to connect it to, there is a lost knowledge. So that's the reason why also museums are so important, is because they keep a lot of these specimens, not just because, oh, we want to collect everything, but it is genuine knowledge. It is knowledge that is place-based in these islands. And so, yeah, so the number here, the number of described species that we had in Hawaii, unfortunately, these are the percent of which they are extinct. And there are some whole families that we have yet to find a living representative in these islands. So we've been trying to fill these knowledge gaps to see what is really left of Hawaiian land snails. And so there are still a lot of questions and identifications that we're trying to do. And so with the help of the technology that Ken was talking about, it has helped us advance much more quickly the diversity that we have here, try to figure out what it is and how they're related to each other. So we have over 1,500 sites that we've been to, but as you can see, there's a lot of areas that we have yet to see and try to survey. Early last September, this past September, we were on Kauai and we rediscovered 2 species of snails that haven't been seen for 100 years. So we're pretty excited that there's still diversity out there that people have counted out as gone, but they're still trying to hang on until we can find them again. We use a different technology approaches, whether it's genetics. We also do SEM imaging, microscopy, and all of these things have helped us try to figure out what we have and how they're related to each other. And so here are a couple of our species rediscoveries. These were on Molokai. These haven't been seen since the early mid-1900s. This beautiful snail here, Armata, is endemic to the Big Island. And this is a snail that Ken had been trying to find for about 20 years and was found last January. So it is still alive. It laid eggs, and to stress Jan out, it takes about 3 months for it to hatch. That's the reason why they're going extinct, right? Because back in the days, there weren't any much predators except for birds. What are the native land mammals that we have in Hawaii? A bat. That's it. That's it.
Do you have a bat here?
Yeah, we have a native bat. Yeah, we do. So all of the pigs, rats, ungulates that you see, they're all invasive. So they didn't have a lot of the predatory pressures of a lot of areas throughout the world. So they didn't have to make babies fast. They didn't have to make a lot of babies, right? Because mostly the rails and other birds were its primary predator, and the carnivorous caterpillar, right? So they could take their time to reproduce and get older before they pop a baby. However, with all of the invasive species that we have now, they haven't had the time to evolve and have a chance to adapt to be able to survive with all of these invasive species here now. And here are 2 other species that we have recently named. And it's not because, oh, we get to name species, it's so much fun. it has a conservation practicality. Because you can't put this on the IUCN Red List, you cannot get any funds for it unless it has a name. So in some ways, when we give it a name, we give it life, as in we give it recognition. And by doing so, that's how we can garner support from the community to try to give some funds to try to save some of these snails. This one here is the last endodonta species on Earth. All of the other ones are extinct. Guess which island this snail is endemic to?
Here.
Not Oahu. What other island? Not Maui, go north.
Nope.
Not south, Big Island is south, north. So we have Maui, Oahu, what's north of us? Kauai? Alright, go more north. Go more north. Close Nihoa. Right? That's the only place in this entire world that still exists on. And we are lucky to actually have a colleague that actually went to Nihoa a couple of weeks ago, brought back 20 or so live of them, and we're gonna have it in captive rearing, so you can come to the museum to see the last endodonta species on Earth, and we're gonna see if we can keep them alive. So, all right. So moving on, Ken, you wanna go through this before we hit our?
Sure. Real quickly, so
Yeah.
just to give you some examples of the use of technology.
And I can put these up.
You know, a lot of times people are like, yeah, I don't really like snails. That's okay. We use snails as a lens to better understand our world because everything's connected. But I wanted to give you a few examples of technology. I'm just going to go back really quick and emphasize something. So, how do we find these, right? Nori mentioned that we looked for this one 15 or plus years looking for it, right? Same thing for these others. How do you think we went about finding it? Like, I mean, Hawaii's small, but so are we. Like, I mean, imagine trying to find a snail, and this snail, you can see that egg is about, what do you think, about third of a millimeter in diameter.
So
It's
that's
tiny.
like half a rice grain.
Yeah, and so this guy is only a few millimeters, right? So imagine finding that on the Big Island. How do you go about narrowing down where you look? Right? Technology can help, right? We use historical samples. People had collected these and given them names before. And they didn't have GPS back when these were collected. Like I said, these were collected the last time they were seen in the 1940s, right? They said where it was at, but it didn't have GPS coordinates. So, I mean, it said Kona. Okay, that's a pretty big area to look for something a few millimeters. Well, we can use GIS technologies, right? So you can learn to do GIS mapping, and that's what we do. We map a lot of these things out. We use some sort of estimating technologies to figure out where the centroid of these areas are, and then develop grid search patterns or plans to search those areas again in the types of habitats. All of that requires technology to digitize that information, sort it, analyze it, use coding to screen it, all kinds of things. And that's how we end up finding these. And then also there's a lot of luck, right? You can read this story if you want, that it's online, about how much luck was involved. But some of the other technologies, like I mentioned, we use genomics, right? And we don't just use it to identify snails, but we also use it to understand. So anybody know what this is? Anybody ever seen these snails before? That is an apple snail. So these are ampullarids. They were introduced to Hawaii for food, but they've become a major pest. They reduce taro production by about 40%, right? So that's a huge hit. They also carry diseases and such, but this is one from the Big Island. You can see all of its little bright colored pink eggs. They're actually kind of cool evolutionarily. They're bright pink because they're telling you don't eat meat. Why? Because they're neurotoxic. They contain a protease inhibitor. It's the only known animal to produce a protease inhibitor. Protease inhibitors are usually produced by plants. So basically what that means is if you eat their eggs, and eggs are usually nutritious. Like if you're going to be a predator or something, you want to eat its eggs first. Why? Because that's where all the nutrition is. Except for these guys, nothing eats them. Why? Well, if you eat them, you get a little neurotoxic, so it won't kill you, but it'll numb you. You'll develop neurotoxic symptoms like tics, muscle control, spasm, things like that. But more importantly, you won't get any nutrition anyway because it prevents you from digesting it, right? So you would just get, it might be a good weight loss drug, who knows? But really interesting, but evolutionarily they're also interesting. Why can't we get rid of apple snails? They're aquatic snails and they're in our taro. We want to get rid of them. Why don't we just dry it down? Just remove the water and then they'll die, right? They're aquatic snails. Well, turns out it's not that simple. Apple snails have been around for about 160 million years. So they kind of know something about surviving. And it turns out they have both the lung and a gill, so they can breathe in and out of water. Not only that, you can take an apple snail, some species, particularly this one, take it out of water, dry it off, wrap it up with a paper towel, and stick it in a jar with no water, and leave it there for years. Come back, put it in water, within about 30 minutes it'll crawl around, be eating and mating. They're incredibly resilient. So there's some really amazing things. So we're using genomics to look at how do they do this, right? Genetically, how do they do that? So it has both biomedical implications, but also implications for how we control them. So we want to get rid of them. Maybe we could modify genes to prevent them from being able to survive desiccation so we could dry them out of the taro vacuum. So there's lots of really interesting technology to be applied to that. Same thing with our Hawaiian land snails. So why? Out of 759 species, we have roughly 320 that we know of. Probably a few more than that because we keep finding them. But roughly 60% are gone. So that 45%, how did they survive and why? Well, maybe it's in the genes. Maybe there's something genetically different about them and how they live and how they express themselves in changing climates and things. So we're using this to sequence the genomes of these snails as well to better understand. And then like I mentioned, so what do Hawaiian land snails eat? Anyone know? Anyone? Nope, me neither. Believe it or not, we've been studying land snails for captively rearing them in Hawaii for more than 30 plus years. We've been at the museum for 20. We still don't know exactly what they eat. We feed them stuff and they survive. They reproduce. We've got what, 12,000 or so now, 42 species. We got a lot of snails in there, but we don't know exactly what they eat. So we just feed them a lot of different things, mostly fungus. But here's the reality is now we have technology, right? And what you see here is happening is who knows what phyllosphere means? Right? Scientists love terms to encapsulate complex ideas. And this was not all that complex. So growing on plant surfaces are microbes, right? See plants, there's fungus, looks like fungus. Most of the time that's cyanobacteria, but it looks like fungus. So they're microbes. Algae, bacteria, fungi, cyanobacteria, all those things growing on plant surfaces. And this is what Hawaiian land snails eat. They're scraping that, most of the ones that are living on plants, they're crawling around scraping that stuff off. They're not eating live plant material, which is kind of cool because most snails in the world eat plant material, right? They become pests, but our snails don't do that. But what is it they're eating? So that's a really hard thing to tell, right? If you know anything about microbiology, you can go out and try to culture stuff, right? But there may be 10 trillion bacterial species in the world, and most of them we can't culture, and most of them we don't know. But again, to get it that quickly, we can go out, and what you see them doing here, this is an undergraduate from Pomona College that came out last summer, and she's swabbing a plant. And what we do is we figure out where the snails are living, which plants they prefer, we swab them all, and we start making graphs like this from the DNA that we sequence them. And what this is just a spiral diversity graph, right? So the big circle in the middle, all bacteria, right? Then we go out, they all belong to this order Pseudomonodonta, right? And they're alpha protobacteria, and this getting further out here gets you down to a lower taxonomic level, species, things like that. So look at how much diversity there is, right? And this is just one sample and one order. You can do this with each of the different orders. And it turns out there's 1,000 species of different bacteria on each of these plants and 1,000 species of fungi. And they're in different proportions, and it turns out they're in different proportions on different plants. It turns out the plants that the snails prefer to live and eat on have a different community of microbes than those that they try to avoid. What's driving that? So that's the next question. So we're trying to figure all of that out using genomic techniques, right? You can see here swabbing away at their leaves with the burns in the forest. Right. Next up.
And so now we're going to go into some of our activities. If you do want to have a quick guide on some of the more common introduced land snails in Hawaii, as well as a quick guide to Hawaiian land snail family. So this is one of the guides here that you guys can see. Just click on this QR code, and then all you have to do is put in your name, what you're going to be using it for, and then you guys can download some of these guides that we actually have printed over here also. So if you guys want to take a look at it, here are some of the guides that we made. So these are some of the things that you are a graphic designer, graphic artist. We would love to have you help us create more of these type of content for us. Everyone got it? It's also, this is a link also, if you guys want this image, this slide, that you guys can go to. If you want to help with captive rearing, here's a QR code for that. Or just talk to Jan. She's going to be at this table. So we take all types of volunteers. They can either be just volunteer where there's no projects or any other type of things tied to that. You just want to help save our snails, we would love to take you. But if you also want to put this as part of a class project or for experience, please let us know. We have a lot of snails and some field work that you guys can do to help us collect some plants for captive rearing. Everyone got a chance to get that. But again, if you guys don't have the QR code, just talk to Jan and give her your contact, and then we can follow up with you guys. And then the next one, so this is a project right now that we are also taking some interns so that they can get some experience on identifying snails, but also like Ken was saying, trying to figure out what type of plants are needed to have our snails survive. And then this is our two young boys, so that hopefully you guys have a little bit more appreciation of some of our land snails in Hawaii, because we'll only say what we know about, right? And if we only think that they're gross, junk, and not useful for us, no one's really going to care about them. So hopefully after today's session, you have a little bit more appreciation for some of the native species that we still have left in these islands. Okay. So very quickly, Jana again is going to be here for the captive rearing. Ken is going to be at this table to talk to you guys more a little bit about our land snail. He's the one who did all the 3D printing for that. And then I'm going to be on this table and trying to see if you guys can ID some land snails just based on some overall morphological characteristics. And if you guys pass my test where you get at least 75% correct, you guys got some Hawaiian land snail stickers that I'll give you. And so we'll go over some of these type of share characters when we're on this table with me. So if you guys want to learn how to start IDing snails, please be at that table. And again, these are all parts of some of the field guides that we have with that QR code. It has some of these different type of terminologies and stuff, because if you're going to be asking us to ID snails, right, it's going to be helpful if you guys can say, oh yes, beehive, or it's a cinnamon bun, or something like that. We also have field guides over there to show you how to image snails. Okay, you guys don't have Polaroids or photographs that you have to develop. You have smartphones. So please send me non-blurry picture of a snail, because if you do that, then I can't help you at all. I can just say it's a blurry brown thing. So that's some of the things that we can teach you at the museum if you are interning with us. And with that, we have a lot of different agencies and collaborators that we have worked with over the years. And with that, we can start doing our interactive. Yeah, sounds good? Okay.
Yeah, so before we disperse too much, the surveys that you guys have, if you could take a few minutes and do the final page of that, and then you can give it to me or Tatum, or you can leave it at the front desk here. Leanne will collect them. Just make sure they end up with somebody, and that would be fantastic. And if you get a sticker from Nori for identifying things, write your name down. I'm going to put a piece of paper over here, because your name's going to go into a raffle for $100 stipend also. So come try to get some points with her, and enjoy. And please eat more food. There's lots of food. So sign the clipboard here, and please eat all of the food. All right. It's more.